Hello, I'm Stephanie, and welcome to ChromoTek's webinar, Single Domain Camelid Antibody or Nanobody Tools for Research, as written by Dr. Andrea Buchfellner and Dr. Larissa Yerlova. The goal of this presentation will be to introduce you to the advantages of camelid antibodies. This slide shows you the outline of the presentation. First, we'll talk about the source of our antibodies and their special features. Then how we select and produce our alpaca antibodies, and then an overview about the alpaca antibody-based applications. So why are we using alpacas as an antibody source? I'm sure you're familiar with conventional IgG antibodies from rats, mice, and rabbits. They're large, about 150 kilodaltons in size, and they consist of 2 heavy and 2 light chains. Over 20 years ago, a small group of scientists in Belgium were investigating the immune system of dromedaries. They found that dromedaries and all other camelids possess a special subclass of IgGs devoid of light chains. It's not really known why camelids have these special heavy chain antibodies, but it has been found that heavy chain antibodies are as specific as conventional antibodies and in many cases more robust. At ChromoTek, we isolate the antigen binding fragments of the heavy chain antibodies. They're called VHHs, variable heavy chain domain of heavy chain antibodies, or referred to as nanobodies. They're small, only 13 to 15 kilodaltons in size. So, what's so special about these VHHs? In addition to their size, a VHH is just a single polypeptide chain. They're highly stable and soluble. They have a high affinity to their antigen, and their single domain character makes them very suitable for recombinant expression in heterologous systems like E. coli. VHHs have an extended complementarity determining region 3, or CDR3, compared to variable heavy domains of conventional antibodies. This extended CDR3 allows them to reach into pockets and clefts of enzymes. Here, you can see a 3D structure of a VHH in complex with a hen-egg lysozyme. The CDR3 is marked in red, and you can see that the VHH reaches into the enzyme's pocket with its finger-like CDR3. There are several publications describing VHHs, which are able to inhibit or modulate the enzymatic activity. How do we select and produce our nanobodies? The process from start to finish takes roughly 4 to 6 months. Here you can see the workflow. We start with the immunization of the animal using the protein of interest. We then isolate RNA from the lymphocytes, do cDNA synthesis, and then clone the VHH sequences into a vector to create a large and diverse phage library. For selection of specific nanobodies, we use phage display. Once we identify positive clones, we have to characterize each VHH. ChromoTek focuses on the generation of nanobody-based reagents, and the next slides will provide examples of the advantages of using nanobodies for applications like immunoprecipitation, super-resolution microscopy, live cell imaging, and live cell detection of protein-protein interactions. I'd now like to show you the advantages of using nanobodies for IP. For that, we couple nanobodies to different solid supports, creating so-called Nano-Traps. Nano-Traps are suitable for many kinds of applications, including proteomic studies, enzyme activity measurements, chip-RIP analysis, or you can couple the nanobody on a chip surface for affinity measurements. The first trap I want to introduce you to is our GFP trap. It's based on a nanobody directed against the green fluorescent protein for immunoprecipitation of GFP-tagged proteins. GFP is a widely used tag and enables you to see where your fusion protein is localized in the cell, and you can check expression levels of your protein. Here, you can see the comparison of the GFP trap with a conventional anti-GFP antibody immobilized with protein AG. You see Kumasi blue stain gels, and the loading is input, flow-through, and bound fraction. An immunoprecipitation of GFP from HEC293 T cells was performed, and bound protein complexes were eluted with a denaturing SDS sample buffer. For the GFP trap, you can see one clear single band in the bound fraction and almost no background, whereas for the IP with a conventional antibody, you see two additional bands, which are the heavy and light chain of the antibody used to detect the GFP. The GFP Nano-Trap enables purification of your protein of interest without contamination of heavy and light chains.
We've picked one example for an interesting application of the GFP trap carried out by one of our customers for a ubiquitination assay. He co-transfected a GFP-tagged protein together with flag-tagged ubiquitin. For ubiquitination assays, you sometimes have to wash with harsh buffers to remove unspecifically bound material with high concentrations of urea. In these experiments, it was possible to wash the samples with 8 molar urea without compromising the binding of GFP trap to GFP. The customer was able to identify ubiquitination of 6 candidate proteins in flybrain during development. The GFP trap recognizes derivatives of GFP like M-clover, YFP, Venus, fluorin, and many others. We've also developed a nano-trap directed against red fluorescent proteins called our RFP trap, which also recognizes many variants of RFP like M-cherry, tag RFP, and so on. The GFP trap is used in more than 660 publications. Since the GFP nanobody is directly coupled to the beads, the trap is ready to use, and you can go from cell to gel in 30 minutes. The GFP trap has a very high affinity to GFP in the picomolar range and is compatible with most wash buffers. As I already mentioned, it's functional under harsh conditions like 8 molar urea over a wide pH range and high salt concentrations. You can elute the bound protein complexes with SDS buffer or glycine, but you don't need to elute. You can use the beads directly in your downstream applications. You can send the beads to a mass spec facility for on-bead digestions, or you can do enzymatic activity tests.
The next nano-trap I want to show you is our GST trap. It's a nanobody directed against glutathione S transferase. GST is also a bigger protein tag at 26 kilodaltons in size. But because GST rapidly folds into a stable and highly soluble protein upon translation, insertion of the GST tag often promotes greater expression and solubility of recombinant proteins. We performed an immunoprecipitation of GST from E. coli cells. You can see a Kamasi blue stain gel, and the loading was input, flow-through, and bound fraction. We eluted bound GST with SDS sample buffer. You can clearly see that there is one single band in the bound lane. Another tag trap I want to show you is our MIC trap. You can see here the MIC trap compared to a conventional anti-MIC antibody immobilized via the protein AG. We performed an immunoprecipitation of a MIC-tagged protein from HEC293 T cells. The loading was input, flow-through, and bound fraction. The bound protein complexes were eluted with SDS sample buffer. Again here, with the nano-trap, you get one single band, whereas for the IP with a conventional antibody, you will get 2 additional bands, the heavy and light chains from the antibody you're using for your detection. The last nano-trap I want to talk about is directed against an endogenous protein. It's our PARP1 trap. The trap is directed against the nuclear protein poly-ADP ribose polymerase 1. PARP1 is a nuclear protein involved in DNA repair. When you induce DNA damage, PARP1 gets recruited to the site of the damage and starts to attach long-branched poly-ADP ribose chains to itself, to other proteins, and to the DNA. The PAR chains are negatively charged, and due to the negative charge, the DNA starts to relax, and the DNA repair machinery can get access and do its job. We immunoprecipitated endogenous PARP1 from HEC293 T cells. You can see a Kamasi blue stain gel on the left and a Western blot on the right, which was probed with an anti-PARP1 antibody. The loading is input, flow-through, and bound fraction. We could successfully precipitate the endogenous PARP1 enzyme, and then we wanted to know if PARP1 is still enzymatically active. For that, we performed an on-bead perylation assay. After one-step immunoprecipitation of endogenous PARP1, we added a reaction mixture, including NAD+ as a substrate, histone 2A as an acceptor for poly-ADP ribose moieties, together with a double-stranded oligonucleotide that mimics a DNA strand break. You can see here that on the silver gel, we successfully synthesized and extracted PAR chains of different sizes. So, the immunoprecipitated PARP1 retains its enzymatic activity and can be used for perylation assays. Here's an overview of the currently available Nano-Traps. I've already introduced our tag traps to you. We are launching an MBP trap very soon, which is a nanobody directed against maltose binding protein. But we also have other gene-specific nanotraps, like against DNMT1, 2 against P53, both at the N and C terminus, MDM4, and MK2. We coupled our nanobodies to different matrices like agarose beads, magnetic agarose beads, and magnetic sapphirose beads. Now I want to give you some tips and tricks on how to get your IP or co-IP to work. The first step is choosing a good antibody. Please keep in mind that not all antibodies work with IP. Good working antibodies are available against protein tags like GFP, MYC, or FLAG, and I always recommend to use antibodies that are validated in IP and co-IP, or use antibodies that are published or certified. The next step is choosing your matrix. Keep in mind that every matrix will have some nonspecific binding because there are no inert surfaces or matrices. There are databases available like crapome.org, which can help you determine the background contaminants, for example, proteins interacting with your solid phase support, the affinity reagent, or the epitope tag that you're using. Then you have to think about how to immobilize the antibody. You can use protein AG beads for non-covalent immobilization, but the antibody will come off the beads if you elute with SDS sample buffer or glycine. You can also cross-link the antibody, or you can use NHS coupling to couple the antibody to your solid support. Please choose the right controls for your co-IP experiment. Check to make sure your endogenous or target protein does not stick to your matrix. The next step is preparing the cell lysate. You can use a non-denaturing lysis buffer, which is suitable for co-IPs, or you can use RIPA-Buffer. It's a harsher lysis buffer that contains more detergent and is typically used to release nuclear proteins. Again, keep in mind that RIPA-Buffer can denature your proteins or may disrupt your protein complex of interest. If you want to immunoprecipitate your protein, it has to be soluble. So please also check your pellet fraction in case your protein isn't soluble, and then you have to change the buffer conditions. To prevent degradation of your protein, keep your samples on ice and add protease inhibitors. That's very important when you're working with small peptide tags like MYC, FLAG, or HA because they don't have a secondary or higher order structure. If you want to preserve post-translational modifications of your protein, you can add phosphatase inhibitors. The next step is the immunoprecipitation. Incubate your antibody coupled to the matrix with your cell lysate. Then remove the non-bound fraction and wash the beads to get rid of all the background. The salt and detergent concentrations during IP and washing depend on how weak or strong your proteins interact. You can also play around with incubation times and volumes. In the last step, you have to decide if you want to elute your bound protein complexes and how you want to elute. It depends on your planned downstream application. You can either use a denaturing elution buffer like SDS sample buffer, a non-denaturing buffer, or high salt. If you don't have to elute, you can perform your downstream application directly on the beads. Now let's look at the advantages that nanobodies provide for microscopy. When you perform conventional immunostainings, most of the time you use a primary and then a secondary antibody. This is called indirect immunofluorescence. At ChromoTek, we have developed specific reagents for direct immunofluorescence by conjugating our nanobodies directly to fluorescent chemical dyes and creating so-called nanoboosters. We developed nanoboosters for immunostaining of fluorescent fusion proteins. Our GFP booster specifically stains different green fluorescent protein variants such as CFP, GFP, EGFP, YFP, Venus, TAG GFP, fluorine, and M clover. With our RFP booster, you can immunostain the most common RFP derivatives such as M cherry, photoactivatable M cherry, M plum, M RFP, M RFP ruby, and D tomato. We conjugate our nanoboosters on bright and stable fluorophores with a stoichiometry of approximately 1 to 1. You can choose between green ADO 488, red ADO 594, far red ADO 647N, or a barrier star 635P. With GFP and RFP boosters, you can do direct immunofluorescence in 1.5 hours from fixation to imaging, since you won't need secondary antibodies. On the right-hand side, you can see an overview of applications that the nanoboosters can improve, including immunofluorescence, immunocytochemistry, and immunohistochemistry. What do you gain when you immunostain your samples with nanoboosters? 1. Nanoboosters will increase the signal of your fluorescent proteins. Please note that signal amplification you can achieve with direct immunofluorescence is lower than what you can potentially reach with indirect immunofluorescence. At the bottom of this slide, you can find a quantification of the signal increase with the GFP booster showing about a 2-fold signal enhancement. Nanoboosters will stabilize your fluorescent signal against bleaching. It is often the case that fluorescent proteins, especially red fluorescent proteins, bleach rapidly during imaging. The quantification below shows that RFP signal is much more resistant to photobleaching if it was stabilized with RFP booster. 3. With nanoboosters, you can regain the signal of your fluorescent protein if it was lost due to a chemical treatment of your cells. In situ hybridization or clicket chemistry can kill the fluorescence of your proteins, but you can regain it with a booster. Next, due to their small size, nanobodies show much better tissue penetration than traditional IgG antibodies, which are roughly 10 times larger. And last but not least, multiplexing with nanoboosters is much easier due to the camelid origin of the nanobodies. You can easily combine mouse antibodies, rabbit, and alpaca nanoboosters without having to worry about cross-reactivity. If you do immunofluorescence with nanoboosters, you have a wide range of options for your fixation reagents. GFP and RFP boosters work on samples fixed with 4% PFA, 0.2% glutaraldehyde, 100% methanol, or even after acetone methanol fixation. Furthermore, with nanobodies, you can achieve better structural preservation of your samples. This is especially evident on the following example. If you want to immunostain yeast subcellular structures with conventional antibodies, you will need to enzymatically digest the yeast cell wall. Cell wall digestion, however, leads to a collapse of cell shape, which can in turn damage or even disintegrate intracellular structures. Nanobodies can penetrate the yeast cell wall, making cell wall digestion unnecessary. Immunostaining procedures can go faster, and the structures are better preserved. Here, figures E and F show much clearer vacuolar structure in the yeast cell than what is visible on C and D. And now we come to the greatest advantage of nanoboosters. This is their improvement of image resolution. Resolution of conventional light microscopy is limited to 200 nanometers, but recent advances in microscopy made it technically possible to achieve 10 nanometer resolution. This should allow us to resolve small subcellular structures such as synaptic vesicles or microtubules. However, in order to image them, we should fluorescently label them. Here comes the bottleneck. Conventional antibodies are simply too large. On this slide, we show a very impressive size comparison of a reconstructed synaptic vesicle and of possible affinity detection molecules. It is obvious that the complex of primary and secondary IgGs is about half a diameter of the vesicle, but the nanobody, labeled as VHH here in pink, is significantly smaller. The table next to this cartoon shows us the numbers. IgGs are 150 kilodaltons and 15 nanometers in size, whereas VHHs, or nanobodies, are the smallest antibody fragments, at just 15 kilodaltons and 3 nanometers in size. They enable you to use the full super resolution potential of your microscope. With this slide, we illustrate the high precision of immunostainings that can be achieved with nanoboosters. The cartoon on the left-hand side shows a primary and secondary antibody. The primary antibody binds to the target, the secondary carries the fluorophores. It's clear in this case the fluorescent signal is 20 to 30 nanometers away from the target. Next to it, you can see a tiny nanobooster labeled as VHH/nanobody. The nanobooster brings fluorophore much closer to the target. There's just about 3 nanometers between the target and the fluorophore now. In the middle, you can see a cartoon of the cross-section of a microtubule stained with conventional antibodies on the top, with single-chain antibodies in the middle, and with a nanobody at the bottom of the cartoon. It's obvious that the conventional primary-secondary antibodies make the microtubule appear almost twice as large. Nanobodies, in contrast, add just a few nanometers to the width of the microtubule. The quantification of this effect is shown on the right. Clearly, the width of the microtubules stained with the GFP booster is much closer to the actual thickness of a microtubule, shown as a green dashed line. Here we want to show the application of nanoboosters in direct stochastic optical reconstruction microscopy, or DSTORM. Nanobooster stained structures in mammalian cell lines, primary neurons, and yeast have been successfully resolved with one-color storm. For example, with GFP booster, Helga Evers and Christian Winterflood could determine the nuclear pore diameter. Our GFP and RFP boosters have been applied by a group from Rockefeller University to study spatial organization of escort protein machinery with two-color storm. You can see the images from their manuscript at the bottom of this slide. To optimize our nanoboosters for superior performance in stimulated emission depletion, or STED, microscopy, we conjugated our GFP nanobody to the extremely bright and stable Abirior star 635P dye. You can compare confocal and STED images of Vimentin filaments, where Vimentin GFP is stained with the GFP booster conjugated to Abirior star 635P. Nanoboosters have also been successfully applied in structured illumination microscopy, 3D SIM, and in ground state depletion microscopy, or 3D GSD. Here, you see the images of subnuclear structures, labeled with the DNMT1 GFP and GFP booster, and of an intracellular bridge structure, labeled with CHIM4B GFP and GFP booster. These were acquired with a wide field microscope, or 3D SIM. The next topic is live cell imaging with nanobodies. This is something that's almost impossible to do with conventional antibodies on intracellular targets. In contrast, with nanobodies, you can easily visualize endogenous intracellular proteins in real time. Our nanobody-based agents for live cell imaging are called Chromobodies. A Chromobody consists of a specific nanobody which is genetically fused to a fluorescent protein. This allows real-time labeling and detection of endogenous intracellular components from different cellular compartments. What is the advantage of using a chromobody instead of directly fusing your target to a fluorescent protein? Let's imagine you overexpress actin GFP. All you will see in the green channel is the signal from the chimeric actin GFP molecules and not endogenous actin. You must hope that the chimeric actin GFP molecules will behave and localize in a similar manner that endogenous actin molecules would. In contrast with our Actin Chromobody, you will be able to monitor and localize endogenous actin molecules. We have developed several chromobodies highlighting important cellular processes and structures. These are the cell cycle, lamin, actin, DNMT1, PARP1, and vimentin. Note that chromobodies can be simply transfected into live cells. There's no need for fixing the cells or for doing protein transduction, which is often tricky or not efficient enough for imaging. Transfected cells will produce fluorescent chromobody in their cytoplasm, and inside of a living cell you will see immunostaining. Chromobodies are available as plasmids for transfection or as stable cell lines. Our first chromobody, the cell cycle chromobody, allows you to monitor progression of cells through G1 phase, S phase, G2 phase, and mitosis. Also, early, late, and mid S phases can be discriminated in live cells expressing the cell cycle chromobody. The chromobody is directed against PCNA, proliferating cell nuclear antigen. The chromobody will highlight the cell body, nucleus, and replication foci. The distribution of the chromobody signal can be easily monitored by fluorescence microscopy. The chromobody signals can be easily quantified using an image analysis software. On this slide, you can see an example of quantitative analysis of the effects of chemical compounds on the cell cycle. HeLa cells stably expressing cell cycle chromobody were seated in 96 well plates and treated with increasing concentrations of acetylcholine, nicotazole, or staurosporine for 24 hours. The cells were imaged and quantitatively analyzed in an automated fashion. Cell-by-cell analysis was based on nuclear size, shape, intensity, and the number and size of nuclear granules or spots, representing replication foci. As you can see from the plotted data, treatment of cells with acetylcholine, which is a DNA polymerase inhibitor, increases the number of cells stuck in early S phase. Treating cells with nicotazole, a microtubule depolymerizing agent, increases the number of cells stuck in mitosis, since the metaphase spindle can't form. Staurosporine treatment increases the number of dead cells by inducing apoptosis. In this way, the cell cycle chromobody allows you to obtain detailed, multidimensional, multiparametric data from one single assay. The Lamin Chromobody visualizes nuclear lamina. With the Lamin Chromobody, you can highlight the nuclear envelope, determine nuclear size and shape, or monitor the disassembly and reassembly of the nuclear envelope during mitosis. Fragmentation of nuclear lamina during apoptosis can also be monitored in real time. Here you can see U2OS and HeLa cells stably expressing Lamin Chromobody, and how the Lamin Chromobody signal changes throughout the cell cycle. The Lamin Chromobody is also suitable for high content analysis. Here we analyzed the time course of apoptosis in cells treated with increasing concentrations of staurosporine. Live HeLa cells stably expressing the Lamin Chromobody were imaged for 20 hours during incubation with staurosporine. Fragmentation of their lamina was quantitatively evaluated and plotted on this graph. As you can see, the 5 micromolar staurosporine curve reaches its plateau after 12 hours of incubation. The Actin Chromobody visualizes the actin cytoskeleton and its dynamics in live cells. The Chromobody highlights endogenous cellular actin and shows localization and reorganization of cellular microfilaments. Here you can see HeLa cells expressing the Actin Chromobody. The images seen are cells during cytochalasin D treatment, which disrupts actin microfilaments. The cells were allowed to recover from the treatment, and you can observe how the microfilaments repolymerize. The Actin Chromobody is functional inside various cell types from different species, and our customers have published papers using this Chromobody in human, mouse, hamster, tobacco leaf cells, and zebrafish. Recently, a transgenic zebrafish expressing Actin Chromobody has been generated. The fish is viable, nicely fluorescent, and the Actin Chromobody does not seem to interfere with fish physiology. Recently, a nuclear Actin Chromobody has been generated and applied to study F-actin assembly and distribution in cell nuclei. DNMT1 Chromobody enables visualization of endogenous DNA methyltransferase inside live cells. DNA methylation is an important epigenetic marker regulating gene expression. DNMT1 functions during DNA replication to copy the DNA methylation pattern from the parental DNA strand onto the newly synthesized daughter strand. During the S phase of the cell cycle, DNMT1 shows punctuate distribution in the cell nucleus. The DNMT1 Chromobody visualizes subcellular localization of DNMT1 without affecting its enzymatic activity. PARP1 is our most recently developed Chromobody. It allows real-time visualization of endogenous human poly-ADP ribose polymerase I, an enzyme involved in DNA damage repair. Here you can see a laser microirradiation experiment where PARP Chromobody visualizes that endogenous PARP1 is recruited to the DNA damage sites in human cells. Non-human BHK cells served as a negative control since the Chromobody does not bind hamster PARP1. We're coming to the last application of nanobodies that we'll talk about today. Live cell detection of protein-protein interactions, or PPIs. All cellular processes are orchestrated by finely tuned protein teams. That is why it is extremely important to have robust and reliable assays, which allow us to monitor interactions between the proteins. Based on our nanobodies, we created a PPI platform at a specific location in the nucleus of mammalian cells. This platform will recruit your GFP-tagged bait protein that you can easily see if your RFP-tagged prey interacts by simply checking if it follows the bait to the platform. We call this tethering assay the fluorescent 2-hybrid assay, or F2H. It enables microscopy-assisted detection of PPIs in live mammalian cells. As mentioned, we engineered a BHK cell line to carry an anchor for GFP at a specific location in the nucleus. This means that when a GFP-tagged bait protein is transfected into the cell line, it will be enriched at this platform and form a green spot. If a co-transfected RFP-tagged prey protein interacts with the bait, it will be enriched at the same location, forming a red spot. F2H was designed to be used with already available constructs. Fusions with GFP and popular GFP derivatives are used as the bait, and fusions of RFP or RFP derivatives are used as the prey. You can easily apply F2H to validate your hits or identify PPI domains. Since the assay is fully reversible, you can test PPI inhibitors and observe their inhibitory effect in real time in live cells. To detect the interactions, you use a conventional efflorescence microscope and a 20x objective. There's no need for confocal laser or high-tech analysis. Looking at the image in the bottom left, you see the same cell imaged in both green and red channels. Looking at both channels, you can see co-localized spots indicating that the proteins interact. These proteins are p53 in green and MDM2 in red. If we add an inhibitor to this interaction, we should be able to see how the interaction is disrupted and the red spot disappears. We added a potent stable peptide developed in David Lane's lab to inhibit the p53-MDM2 interaction. And as we can observe, within 2 hours of incubation, the red spot's gone and the interaction is disrupted in live cells. On the left-hand side of this slide, you can see a list of protein interactions that have been validated with F2H. These interactions are with nuclear, cytoplasmic, and even mitochondrial proteins. F2H can be used to identify interacting protein domains. As seen on the right, XRCC1 was used as bait, and DNA ligase 3, both full and partial domains, served as prey. When the full-length proteins were used, you can see that both spots are present, indicating that XRCC1 and DNA ligase interact. When just the BRCT domain of DNA ligase 3 was used, you still see interactions. However, when the whole N-terminus of the DNA ligase 3 lacking the BRCT domain is used, interaction was lost. You see only one spot, indicating that the whole interaction is confined to the BRCT domain. With F2H, you can define what domains of your proteins are responsible for the interactions. I also want to mention that it's difficult for full-length membrane proteins to be translocated to the nuclear PPI platform, but you can use their cytosolic domains and analyze their interactions with F2H. Now looking at another example using F2H. Here we applied F2H to analyze the interactions not between two different proteins, but between two domains of one protein. We did this with our recently developed androgen receptor F2H assay. A short introduction. Androgen receptor signaling is instrumental for developing male sexual phenotypes. Unfortunately, overactivation of androgen receptor is associated with prostate cancer. This is why androgen receptor activation is an important target for developing inhibitory drugs combating prostate cancer. One needs a good assay to screen for inhibitors. As you can see, androgen receptor consists of an N-terminal domain, NTD, DNA binding domain, hinge region, and a C-terminal ligand binding domain, or LBD. An early event in androgen receptor activation is interaction of its N and C termini. As the schematics on the right show, an active androgen receptor is localized in the cytoplasm. When a ligand comes, for example, dihydrotestosterone or DHT, it binds to LBD, induces a conformational change. LBD interacts with the NTD, androgen receptor goes to the nucleus and starts transcription of target genes. There are various reporter assays that look at transcription initiation. However, for screening, it can be useful to look at the early events in androgen receptor activation. This allows to eliminate artifacts from downstream effects of test compounds on the androgen receptor cascade or on transcription in general. This is why we developed an assay to look at NC terminus interactions of the androgen receptor. To analyze this, we split the androgen receptor protein into two independently expressed domains. One was tagged with RFP and the other with GFP. Both constructs were co-transfected into the F2H cells and analyzed by epifluorescence microscopy. On the left side of this slide, you can see live cell time-lapse images of the F2H androgen receptor assay. In the top row, you can see that without the ligand, green spots are visible, but there are no red spots. Upon androgen stimulation, here we applied 10 nanomolar DHT, the red spots appear. The bottom rows show the same 4 cells, now with co-localizing green and red spots. With F2H, you can not only visualize, but also easily quantify the interactions. On the right, you can compare dose responses of the androgen receptor NC interactions to several clinically relevant inhibitors. We pre-incubated transfected cells with different concentrations of inhibitors, then added DHT, fixed, imaged, and analyzed the images. With image analysis software, you can easily segment the nuclei based in the DAPI staining, the spots in the nuclei in the green channel, and then measure whether there is a local increase of fluorescence in the red channel at the locations of the green spots. If there's an increase, that indicates the interaction. From the graph, it's obvious that enzalutamide and bicalutamide were more potent than flutamide or TOK001. Abiraterone was not able to inhibit the interactions in wild-type androgen receptor at all. In the next step, we performed analysis of mutant androgen receptor response to the inhibitors. It's well known the various acquired mutations within the androgen receptor sequence often render the androgen receptor resistant to inhibitory treatments, and some inhibitors can even activate mutant androgen receptor variants. Here, we generated and tested several clinically relevant mutations. We could show that even one small point mutation, such as W741L, can basically reverse the effect of bicalutamide and turn it into an agonist of androgen receptor NC interactions. ChromoTek's portfolio can be categorized into 4 key applications: immunoprecipitation, immunofluorescence, live cell imaging, and protein-protein interaction studies. With our Nano-Traps, you can do high-yield IP in 30 minutes. And should you already have GFP or RFP fusion constructs, keep in mind that you can use them not only for imaging, but also for IP. And with a short, simple experiment, you'll have biochemical data to support your microscopy results. With our Nano-Boosters, you can do direct immunofluorescence of your GFP or RFP tagged proteins to recover your fluorescence signal, or for super resolution microscopy. With Chromobodies, you can monitor where your endogenous target is localized in live cells. And with the F2H, you can quickly detect whether your GFP and RFP fusion proteins interact and whether your inhibitors can disrupt this interaction. If you are looking for VHHs for other applications, such as co-crystallization chaperones or to stabilize unstructured proteins or whole protein complexes, we do offer certain custom products and services, including custom nanobody generation and also F2H compound screening. I also want to draw your attention to the database of publications from our customers available on our website. By now, there's over 800 peer-reviewed research papers where our products were successfully used. In this database, you can search by organism, product, or technique. Select the papers that are relevant for your experiments. Thank you so much for your time and attention. Please do not hesitate to send us an email or give us a call if you have any questions regarding this presentation. I strongly encourage you to check out our website. In addition to the reference database, we also have sections for frequently asked questions, new products, and additional information on our blogs that are updated regularly. Also, we have forums to request free test samples on specific products within our portfolio. Thank you again, and I hope you have a wonderful day.